This is Lecture 17 on Chapter 17, titled From Gene to Protein. As an overview, we note the flow of genetic information is from DNA to RNA, and most RNA will be used for production of protein. The information content of DNA, the genetic code, is in the form of specific sequences of nucleotides along the DNA strands. The DNA inherited by an organism leads to specific traits by dictating the synthesis of proteins. This is what we have alluded to in the prior lectures leading up to this one that focuses on that aspect of living organisms. The process by which DNA directs protein synthesis, gene expression, includes two stages called transcription and translation. And we will define those as we go along. The ribosome is part of the cellular machinery for translation, the polypeptide synthesis. So transcription is from DNA to RNA, and translation is from RNA to the protein or polypeptide or protein synthesis. This illustration is a computer model of a ribosome, part of the protein synthesis machinery. So there are several different parts of it that you see in, represented by all of these different colors. Concept 17.1, genes specify proteins via transcription and translation. Evidence from the study of metabolic defects had led to the idea of genes producing proteins. In 1909, British physician Archibald Garrod was the first to suggest that genes dictate phenotypes through enzymes that catalyze specific chemical reactions in the cell. Later studies examined the nutritional mutants in Neurospora, and the scientific inquiry was performed by Beadle and Tatum, looking at the what causes bread mold to mutate with X-rays, creating mutants that could not survive on minimal media. So the Neurospora, or bread mold, that they studied could live on this minimal media that just had to have a few components in it, didn't have to have the full, protein and carbohydrate and all that stuff. It just needed minimal media, and then it had everything it needed to produce everything else, all the other biological macromolecules required for it to live and reproduce. So when they exposed some to X-rays, they found that there were mutants that required additional components in the media in order to survive. So using genetic crosses, they determined that their mutants fell into three classes, each mutated in a different gene, and we see that in this figure here. The question was, do individual genes specify different enzymes in arginine biosynthesis? So working with the mold Neurospora crassa, George Beadle and Edward Tatum had isolated mutants requiring arginine in their growth medium and had shown genetically that these mutants fell into the three classes, each defective in a different gene. From other considerations, they suspected that the metabolic pathway of arginine biosynthesis included the precursors ornithine and citrulline. So their most famous experiment is shown here. Tested both their one gene, one enzyme hypothesis and their postulated arginine pathway. In this experiment, they grew three classes of mutants under the four different conditions shown in the results here. So the wild-type strain required only the minimal medium. We see here, wild type in this column, and this is the minimal medium control. The three classes of mutants had different growth requirements. So at the minimal media, the class 1, class 2, and class 3 mutants did not grow. With the minimal media plus ornithine, the wild type and the class 1 mutant grew, but not the class 2 and class 3. When citrulline was added, the growth occurred all the way through the class 2 mutants, and then the class 3 mutant was unable to synthesize the arginine even with precursors, and we see that it only grew when arginine itself was provided. The conclusion then, from the growth patterns of the mutants, Beadle and Tatum deduced that each mutant was unable to carry out one step in the pathway for synthesizing arginine, presumably because it lacked the necessary enzyme. Because each of their mutants was mutated in a single gene, they concluded that each mutated gene must normally dictate the production of one enzyme. Their results supported the one gene, one enzyme hypothesis and also confirmed the arginine pathway. So notice that a mutant can grow only if supplied with a compound made after the defective step. So here we see the precursor and enzyme A converting it to ornithine, enzyme B converting that ornithine to citrulline, and then the citrulline is converted to the arginine by enzyme C. And each enzyme has a corresponding gene, gene A, B, and C. So mutant 1 had a mutation in gene A. So when ornithine or citrulline were provided, or arginine, it could grow. The class B mutant had a mutation in gene B, so only when the citrulline or arginine was provided could it grow. And then the class 3 mutant had a mutation in gene C, and could only grow when arginine was provided. Well, Beadle and Tatum developed the one gene, one enzyme hypothesis, which states that the function of a gene is to dictate the production of a single enzyme, a specific enzyme, excuse me. The products of gene expression, a developing story. Well, as researchers learned more about proteins, they made minor revisions to the one gene, one enzyme hypothesis. Genes code for polypeptide chains or for RNA molecules. So not all genes encode for proteins. Some encode only for RNA, particular types of RNA, and those RNA molecules perform other functions, as we will see. Basic principles of transcription and translation are shown on this slide. Transcription is the synthesis of RNA under the direction of DNA, and it produces the messenger RNA, or mRNA. Translation is the actual synthesis of a polypeptide which occurs under the direction of the mRNA, and it occurs on ribosomes. In prokaryotes, transcription and translation occur together. Here we see the DNA being transcribed to mRNA, and the ribosome is attaching to it and producing the polypeptide according to the genetic code in the sequence, and the translation is occurring. We'll see another illustration of this that actually is actual evidence of the coupling of transcription and translation in prokaryotes. So this is a prokaryotic cell. In a cell lacking a nucleus, the mRNA is produced by transcription and is immediately translated without additional processing. So there's no nuclear envelope, there's no processing of the RNA. As soon as it is being made from the DNA, the ribosomes can interact and translate it into proteins. In eukaryotes, the RNA transcripts are modified before becoming true mRNA, or mature mRNA, or functional mRNA, whichever term you want to use. So here, eukaryote, we recognize the nuclear envelope, and the DNA is transcribed to make pre-mRNA. RNA processing occurs to produce the mature or functional mRNA that now has permission to leave the nucleus. Once in the cytoplasm, then it can interact with the ribosomes, the only location of the functional ribosomes. Ribosomes are assembled within the nucleus, but they are not functional until they have translocated to the cytoplasm. So this is a eukaryotic cell. The nucleus provides a separate compartment for transcription. The original RNA transcript, called pre-mRNA, is processed in various ways before leaving the nucleus as mRNA. So the cells are governed by a cellular chain of command, DNA to RNA to protein. We then ask, what is the genetic code? How many bases correspond to an amino acid? Well, there are 3 of them. They're called codons, and they're triplets of bases. So a codon is a triplet of bases. So there's always the joke, oh, I notice you're wearing your jeans today, and you also have your codon. Anyway, the genetic information is encoded as a sequence of non-overlapping base triplets, or codons. During transcription, the gene determines the sequence of bases along the length of an mRNA molecule. So here we see a DNA molecule, the double helix, gene 1, gene 2, and gene 3. And then we zoom in to look at just a small piece of a gene, the DNA strand, the template strand, notice 5' to 3', and transcription occurs producing mRNA that is actually the complement to the DNA strand, and also, of course, other than having a different sugar, the ribose rather than deoxyribose, you find T in the DNA, but not in the RNA. In the RNA, you find the U. We had studied that before, as you recall. So the mRNA is produced. We see that it is anti-parallel and complementary to the template strand. And then translation occurs where each codon, 3 nucleotides in sequence from the code in the DNA to the messenger RNA is encoding 3 at a time, the triplet, the codon, for a particular amino acid residue within a protein. So one amino acid is referred to as a residue. So residue 1, residue 2, residue 3, residue 4, that type of thing. So cracking the code. A codon in messenger RNA is either translated into an amino acid or serves as a translational stop signal. So here we see the table for all the different codons for the 20 amino acids. You notice that there is redundancy but no overlap in those. Let's see. So the dictionary of the genetic code is shown in this table. The 3 bases of an mRNA codon are designated here as the first, second, and third bases, reading in the 5' to 3' direction along the mRNA. The codon, AUG, not only stands for the amino acid methionine, but also functions as a start signal for ribosomes to begin translating the mRNA at that point. 3 of the 64 codons function as stop signals, marking the end of the genetic message. So codons must be read in the correct reading frame for the specified polypeptide to be produced. Evolution of the genetic code, we note that the genetic code is nearly universal, shared by organisms from the simplest bacteria to the most complex animals, including the codons. So there might be slightly different among species, but they are essentially universal. So in laboratory experiments, genes can be transcribed and translated after being transplanted from one species to another. Here we see a tobacco plant that's glowing like a firefly because indeed it is expressing the gene for the firefly enzyme called luciferase. So this is a very useful tool in molecular biology, recombinant DNA techniques that we'll discuss in upcoming chapters. So the transcription is the DNA-directed synthesis of RNA. Let's take a closer look. The molecular components of transcription include the RNA synthesis. It is catalyzed by RNA polymerase, which pries the DNA strands apart and hooks together the RNA nucleotides. Follows the same base pairing rule as DNA, except that in RNA, uracil substitutes for thymine, and of course the RNA has the ribose sugar rather than the deoxyribose. Here we look at synthesis of an RNA transcript. The stages of transcription are initiation, elongation, and termination, and they are shown in this figure. Here we see a double-stranded DNA and a gene here, and the start point is noted right here on the left on the template strand. So from the start point to the end point is called the transcription unit. We see there is a promoter upstream to the start point. So to the left would be upstream. That would be in the 3' direction. And 5' upstream is the promoter on the template strand, so the upper strand would be the template strand. And the end point is down here. So initiation is after RNA polymerase binds to the promoter, the DNA strands unwind, and the polymerase initiates the RNA synthesis at the start point on the template strand. Then elongation occurs. The polymerase moves downstream, unwinding the DNA and elongating the RNA transcript 5' to 3'. In the wake of transcription, the DNA strands reform a double helix. And then third, termination. Eventually the RNA transcript is released and the polymerase detaches from the DNA, and the completed RNA transcript, the primary transcript, is now available. There's an error on here. They've got RNA, rewound RNA, that should be rewound DNA, closing back up behind the transcription event. Now, RNA polymerases can continually come in and start when one gets into the elongation stage. The initiation can occur, so you can have very rapid transcription to produce RNA. So here we see the 3' and the 5' end, the 5' and the 3' end, and the direction of transcription is downstream from the start site, and we see that the RNA polymerase will create the nucleotide bond between the nucleotides that are coming in, and they have an affinity for their pair, their mate, and they stick to that, and then the enzyme will ligate it and form the polymer chain of the RNA. So the template strand of the DNA is shown here, and the enzyme is assembling the RNA, and that is the complement of that. And notice that it basically matches the other strand. What, they have an E there. Good gosh. And a G there. Look at the mistakes in this thing. This should read just like this. C-A-U, no T, right? And then C, C, A, and rather than a T, a U, rather than a T, a U. So somebody made some typo errors in there. Again, I haven't been taking the time to correct these before presenting them, but it's good because it challenges us to recognize errors. And the textbook figure is correct. So RNA polymerase binding and initiation of transcription. The promoters signal the initiation of RNA synthesis. The transcription factors, proteins that bind to DNA and affect transcription, help eukaryotic RNA polymerase recognize promoter sequences. So we see that here in this figure. Up in the left-hand corner, we see the inset just showing the eukaryotic nucleus with the DNA being transcribed to the pre-mRNA, RNA processing to produce the mRNA, and the cytoplasm translation occurring. In the first panel up here, what we see is the promoter region upstream of the template strand of the DNA and the start point for that. The eukaryotic promoters commonly include what's called a TATA box, a nucleotide sequence containing TA, about 25 nucleotides upstream from the start point. So here's the start point, so upstream of that to the left. So by convention, the nucleotide sequences are given as they occur on the non-template strand. So second here we see several transcription factors, one recognizing the TATA box and must bind to the DNA before RNA polymerase II can do so. So in eukaryotes, there are three types of RNA polymerase, 1, 2, and 3. RNA polymerase II is responsible for producing messenger RNA. Then on 3 here, we see that the additional transcription factors, the purple structures in here, bind to the DNA along with RNA polymerase II, forming the transcription initiation complex, and the DNA double helix then unwinds, and the RNA synthesis begins at the start point on the template strand, shown in here. So next then, elongation of the RNA strand. As RNA polymerase moves along the DNA, it continues to untwist the double helix, exposing about 10 to 20 DNA bases at a time for pairing with RNA molecules. RNA nucleotides. Okay. Termination of transcription. The mechanisms of termination are different in prokaryotes and eukaryotes. Concept 7.3, eukaryote cells modify RNA after transcription. Enzymes in the eukaryotic nucleus modify pre-mRNA in specific ways before the genetic messages are dispatched to the cytoplasm. So the alteration of the mRNA ends So each end of the pre-mRNA, both the 5' and the 3' ends, is modified in a particular way. The 5' end receives a modified nucleotide cap. We see that here. A modified guanine nucleotide is added, called the 5' cap. And the 3' end gets a poly A tail. So notice that there's always a 5' untranslated region, and then there's the protein coding segment with a start codon and a stop codon, and then there is the polyadenylation signal in the 3' untranslated region. So only this portion here will be translated once this gets to the ribosome in the cytoplasm. So 50 to 250 adenine nucleotides are added to the end. These two modifications at the ends are thought to give the molecule stability so it doesn't degrade rapidly, and also to assist with getting it permission to get out of the nucleus. The next RNA processing that can occur is RNA splicing. So you split genes and then you splice them together to remove a portion. With RNA splicing, what it does is there's a removal of introns and it joins the exons. So along the length of the eukaryotic gene, you don't have just the DNA encoding the protein. There are intervening segments called introns that are not translated. They don't even end up in the messenger RNA. Again, this is only in eukaryotes. For interaction with the ribosomes. And that's what we're talking about here. You've got to excise those and then splice back together the portions of that represent the exons only of the messenger RNA, and exons within the original gene. So the introns are cut out and the exons are spliced together to give you the coding segment, and then of course there's the 3' untranslated region and the 5' untranslated region. Here's another error. That should be 5'. They have a 3 in there. Just horrible. So this is carried out by spliceosomes in some cases. So here we see the RNA transcript that is the complement of the template DNA strand. So the gene in the template DNA strand has an exon 1, an intron 1, and an exon 2, and so on and so forth, whatever it may be. We're at the 5' end here, the RNA transcript, or the pre-mRNA. There is a protein, small nuclear RNAs, called SNRPs, and we see it is a small RNA, stays in the nucleus, and it's associated with the protein. And there's other proteins as well that interact with the ends of the junctions of the introns and exons to form what's called a spliceosome. So you can see these sites of interaction between the SNRPs and the intron, and then there is a break in the DNA at the junction of intron and exons, and then the two blunt ends of the exons will be ligated together, and of course the spliceosome components will then be freed up. There'll be a cutout chunk, the intron, and then the mature mRNA, will already have its 5' cap and poly A tail. Mature mRNA will now be the linear sequence of the exons, unless there's some alternative processing or alternative splicing of the messenger RNA, which can occur for certain genes and under certain conditions. So ribozymes is the term used for the ribosomes by the authors, just occasionally in the text, but they pointed out because they are catalytic RNA molecules that function as enzymes, and they can splice the RNA. Oops. They don't use it for ribosomes, do they? Sorry about that. So these types of small nuclear RNAs are referred to as ribozymes because they are catalytic RNA molecules that function as enzymes and can splice the RNA. So they're supported by the protein, but it's actually the RNA that is producing the enzyme-like activity. The function and evolutionary importance of introns. The presence of the introns allows for the alternative RNA splicing, as I had told you. That's at least one of the reasons believed for the existence of the introns. So proteins often have a modular architecture consisting of discrete structural and functional regions called domains. In many cases, different exons code for the different domains in a protein. So you can see that if there is an alternate splicing to this where exon 2 was not retained, then you would have only the domains 1 and 3 in the protein. They would be linked directly, and domain 2 wouldn't even be present. So the gene would have, as shown in the DNA here, exon 1, intron 1, exon 2, intron 2, exon 3. Remember, you get the 5' and 3' untranslated region in both of these, the UTRs. Then there would be transcription, RNA processing, translation, and you get the protein with the 3, or the polypeptide with the 3 domains. So concept 17.4, translation, is the RNA-directed synthesis of a polypeptide. Let's take a closer look at that. So the molecular components of translation, a cell translates an mRNA message into protein with the help of transfer RNA. So here's another example of RNA produced by cells that is not used to encode the sequence of a protein. It's used to assist with protein synthesis, but the RNA itself does not encode for the amino acid sequence of a protein directly from the DNA. As you know, that's what messenger RNA does. No need to have two things doing the same job. So the translation, the basic concept is shown here. And what we see is the ribosome in brown and the transfer RNAs that in the cytoplasm will pick up amino acids. There's very specific interaction of a particular tRNA with a particular amino acid. There is no redundancy or very little redundancy for these. Actually, no, there is no redundancy. There's 20 types of tRNAs, one for each of the amino acids. And we can see that they can bind to the messenger RNA interacting with the ribosome and get shuffled along and add to the growing polypeptide. Then the tRNA is separated from the amino acid once the next one comes along, and once it has lost it, it falls off the ribosome complex. So the molecules of tRNA are not all identical. Each carries a specific amino acid on one end. Each has an anticodon on the other end, as we saw in that brief overview. So here the structure and function of transfer RNA is shown. A tRNA molecule consists of a single RNA strand that is only about 80 nucleotides long. We see it here. It's roughly L-shaped once it makes its final conformational adjustments. It is basically like a clover leaf, but there's additional twisting and turning so that it ends up looking L-shaped. And at one end of the L is a triplet, the anticodon, and at the other end is the nucleic acid sequence specific for binding a particular amino acid. So this is the two-dimensional structure, the four base paired regions, and so we see them here, 1, 2, 3, 4 base paired regions internal within the molecule, and 3 loops, 1, 2, 3, are characteristic of all transfer RNAs, as is the base sequence excuse me, of the amino acid attachment site at the 3' end. The anticodon triplet is unique to each transfer RNA type. The asterisk marks bases that have been chemically modified, also a characteristic of tRNA. Here in this view, excuse me, we see the three-dimensional structure, and that it does look more like an L. Here's 1 loop, 2 loop, 3 loops, but it is kind of an L formation with the 3' up here. Follow it down around through all the loops and the 5' there. This is the site specific for amino acid attachment, and then this is the illustration used by the textbook with the anticodon at the opposite end, and this is the 3' side to the left and the 5' on the right. So if we followed the 3' down through, then this would be 3' going toward 5' there. Notice that the tRNA is held together by hydrogen bonds. There is a specific enzyme called an aminoacyl tRNA synthetase. There's one for each amino acid as well. Joins each amino acid to the correct tRNA. So here what we see in the upper panel is an amino acid and ATP. So we're starting to see the energy consumption in protein synthesis. The aminoacyl tRNA synthetase enzyme is illustrated by the white here. And the active site of the amino acid and ATP, the active site binds the amino acid and the ATP. And in step 2, the ATP loses 2 phosphate groups and joins the amino acid as adenosine monophosphate here. And there's the phosphate groups. Comes off first as a pyrophosphate that splits to 2 of the phosphates. Then number 3, the appropriate tRNA covalently bonds to the amino acid, displacing the AMP, the adenosine monophosphate, coming off here. And, excuse me, here we see the activated amino acid is released by the enzyme, and we have aminoacyl tRNA, which means an activated amino acid. So linkage of the tRNA to the amino acid is an endergonic process that occurs at the expense of ATP, and the ATP loses 2 phosphate groups, becoming AMP, adenosine monophosphate. Now let's take a closer look at ribosomes. They facilitate the specific coupling of tRNA anticodons with mRNA codons during protein synthesis. So the ribosomal subunits are constructed of proteins and RNA molecules named ribosomal RNA, or lowercase r RNA. And what we're seeing in this illustration is a computer model of the functioning ribosome, the larger domain and the smaller, the larger subunit and smaller subunit. This is a model of the bacterial ribosome showing its overall shape. The eukaryotic ribosome is roughly similar. A ribosomal subunit is an aggregate of ribosomal RNA molecules and proteins. So we can see within here the messenger RNA and the tRNA molecules, and the growing polypeptide up through here passes through a structure, an indentation in the large subunit protein called the exit tunnel. So this is the prokaryote. So this is occurring in the cytoplasm, but right near to where the the DNA is being transcribed to the RNA, and of course the eukaryote would only be in the cytoplasm. Here the ribosome is shown to have the 3 binding sites for tRNA. There's a P site, an A site, and an E site. So we see the P site is the peptidyl tRNA binding site. The E site is the exit site. The A site is the aminoacyl tRNA binding site. Large and small subunit, and the mRNA binding site is shown here. So this is a schematic model with the mRNA and tRNA. A tRNA fits into a binding site where its anticodon base pairs with an mRNA codon. The P site holds the tRNA attached to the growing polypeptide. The A site holds the tRNA carrying the next amino acid to be added to the polypeptide. Discharged tRNA leaves at the E site. So this one will have its amino acid added to the chain by a peptide bond, which will dissociate the growing polypeptide from this tRNA, which will then move to the E site. This one, once the peptide bond is being formed, will move to the P site, and then of course another tRNA will come into the A site. So building a polypeptide, we can divide translation into 3 stages: initiation, elongation, and termination. Sounds very familiar. So the ribosome association and initiation of translation is discussed on this slide. The initiation stage of translation brings together mRNA, tRNA bearing the first amino acid of the polypeptide and 2 subunits of the ribosome. So what we see here is the first thing that occurs on the left. A small ribosomal subunit binds to a molecule of mRNA. In a prokaryotic cell, the mRNA binding site on this subunit recognizes the specific nucleotide sequence on the mRNA just upstream of the start codon. An initiator tRNA here with the anticodon UAC, shown in higher magnification here. AUG on the message, UAC is the anticodon. Base pairs with the start codon, the AUG. This tRNA carries the amino acid methionine. Next, over here on the right, we see GDP being consumed. So this is protein synthesis is an endergonic reaction and process. So the translation initiation complex is shown here. The arrival of a large ribosomal subunit completes the initiation complex. Proteins called initiation factors, they're not shown here, are required to bring all the translation components together. GTP provides the energy for the assembly. It's not ATP, but GTP provides the energy just like ATP can do. The initiator tRNA is in the P site. The A site is available for the to the tRNA bearing the next amino acid. So the elongation of the polypeptide chain is next. In the elongation stage of translation, amino acids are added one by one to the preceding amino acids. So here we see the P site occupied by the tRNA holding the growing polypeptide, and the amino end. Remember we talked about amino terminus and carboxy terminus. The amino end is the one at the first part of the peptide being produced. So we see here the codon recognition, the anticodon of an incoming amino acyl tRNA base pairs with the complementary mRNA codon in the A site. Hydrolysis of GTP increases the accuracy and the efficiency of this step. So here's the 2 GTP to GDP and PI, and we get the new amino acyl tRNA into the A site, and then next peptide bond formation occurs. It's actually an RNA molecule of the large subunit catalyzes the formation of a peptide bond between the new amino acid in the A site and the carboxyl end of the growing polypeptide in the P site. So remember, this was amino end and this is carboxyl end. This step attaches the polypeptide to the tRNA in the A site. So then we've got to get it shifted over to the P site. In order to do that, we've got to move the one that was just in the P site over to the E site. So the translocation then will occur. The ribosome translocates the tRNA in the A site to the P site. The empty tRNA in the P site is moved to the E site. So what was in A is now in P. What was in P is now in E, and it can exit where it's released at the E site. The mRNA moves along with its bound tRNA, so there's a ratcheting. So there's a new codon presented at the A site with each translocation event. Termination of translation. The final stage of translation is termination, when the ribosome reaches a stop codon in the mRNA. So we see this occurring here. When a ribosome reaches a stop codon on the mRNA, the A site of the ribosome accepts a protein called a release factor instead of a tRNA. The release factor hydrolyzes the bond between the tRNA, this bond here, and the completed peptide now that was in the P site. The tRNA was in the P site, and the last amino acid of the polypeptide chain, and the polypeptide is thus freed from the ribosome. And then finally, the two ribosomal subunits and the other components of the assembly dissociate. Now, we followed just one ribosome on one messenger RNA, but it is possible to have a polyribosome where a number of ribosomes can translate a single messenger RNA molecule simultaneously. So here on this one, we see one coming off, as we just discussed with termination, one beginning the initiation, and 1, 2, 3, 4 involved in the elongation. So a mRNA molecule is generally translated simultaneously by several ribosomes in clusters called the polyribosomes. And here we see an actual transmission electron micrograph that shows a large polyribosome in a prokaryotic cell. Scale bar is 0.1 micrometers or 100 nanometers. So we can actually see the ribosomes along the length of the messenger RNA, and you can see the messenger RNA in spots between them. Quite fantastic, isn't it? So completing and targeting the functional protein, the polypeptide chains undergo modifications after the translation process. So I use the term post-translational processing, and the protein folding and post-translational modifications are what will occur. So after translation, proteins may be modified in ways that affect their three-dimensional shape. Remember the structures of proteins, primary, secondary, tertiary, and quaternary. So this is part of what we're considering here, as well as other types of modifications. So after translation, they're modified in ways that affect their three-dimensional shape. First, we think of targeting polypeptides to specific locations. So there's two populations of ribosomes that are evident in the cells, the free and the bound. We studied this in the chapter on the cell. The free ribosomes are in the cytosol. They initiate the synthesis of all proteins. Proteins destined for the endomembrane system or for secretion must be transported into the ER. So they have signal peptides to which the signal recognition particles, or SRPs, bind, enabling the translation ribosome to bind to the ER. So this slide is showing us these various steps in translation, looking at just one ribosome and one messenger RNA. And there are 6 steps or stages that are described here. So the signal mechanism for targeting proteins to the ER is shown in this illustration. A polypeptide destined for the endomembrane system or for secretion from the cell begins with a signal peptide, little green signal peptide here. A series of amino acids that targets it for the ER. This figure shows the synthesis of secretory proteins and its simultaneous import into the ER. In the ER and then the Golgi, the protein is further processed. Finally, a transport vesicle conveys it to the plasma membrane for release from the cell. We had talked about that in the prior chapter. So step 1 here is the polypeptide synthesis begins on a free ribosome in the cytosol. Remember way back in that chapter I talked about fixed ribosomes, that they were fixed to the ER, and then I had to correct myself because this is the real situation. The free ribosomes interact with the messenger RNA and begin translation as a free ribosome. If a signal peptide is produced, then an SRP binds to the signal peptide, the signal recognition particle. This thing in yellow shown here is the SRP, halting synthesis momentarily. So translation is halted when the SRP binds to the signal sequence, the signal peptide, excuse me. And third, the SRP binds to a receptor protein on the ER membrane. This receptor is part of a protein complex, a translocation complex, that has a membrane pore and a signal cleaving enzyme. So this is the translocation complex here before joining with the SRP. This is after joining with the SRP, and we see that the signal peptide is the first inserted in there, and it actually sticks to it, as we see here. How did that one sneak in there? Somebody's animation is not very good. The SRP then will leave the polypeptide, will leave the complex, and the polypeptide resumes growing. Meanwhile, translocating across the ER membrane, the signal peptide stays attached to the membrane. So we see on the side facing us, this is the endoplasmic reticulum lumen, and the yellow is the cytosol. So the signal peptide stays attached to the membrane, and then the signal peptide cleaving enzyme cuts off the signal peptide here, and then finally the rest of the completed polypeptide leaves the ribosome and folds into its final conformation. And then the ribosome dissociates, as we talked about with the termination step. We're now at concept 17.5. RNA plays multiple roles in the cell. Just a bit of a review here. RNA can hydrogen bond to other nucleic acid molecules, can assume a specific three-dimensional shape, sometimes by forming hydrogen bonds within itself. And it has functional groups that allow it to act as a catalyst. So they are actually molecules that can have some type of biological activity other than just a carrier of a message or a carrier of an amino acid. So this table is showing us the types of RNA in the eukaryotic cell. This column on the left is the type, and these are the functions here. Let's read our way through this. So messenger RNA, or mRNA, carries information specifying amino acid sequences of proteins from DNA to the ribosomes. The transfer RNA, or tRNA, serves as the adapter molecule in protein synthesis, translates mRNA codons into amino acids. Ribosomal RNA, or rRNA, plays catalytic or ribozyme roles and structural roles in ribosomes. So it was ribosomal RNA that they used that term ribozyme for. I'd gotten it a bit confused earlier, but this helps to clarify that. So primary transcript is the term for the RNA. That is the precursor mRNA, if you want to think of it that way. The ribosomal RNA, or the tRNA, before being processed by splicing or cleavage. Some intron RNA acts as a ribozyme, catalyzing its own splicing. Small nuclear RNA, or snRNA, plays structural and catalytic roles in spliceosomes, the complexes of protein and RNA that splice pre-mRNA. And then SRP RNA, good God, is a component of the signal recognition particle, the protein RNA complex that recognizes the signal peptide of polypeptides targeted to the ER. And then small nuclear RNA, or SNO RNA, small nucleolar RNA, or SNO RNA, aids in processing the pre-RNA, pre-ribosomal RNA transcripts for ribosome subunit formation in the nucleolus, within the nucleus. Small interfering RNA, or siRNA, and microRNA, or miRNA, are involved in the regulation of gene expression. So there's quite a bit of diversity of the types of RNA and the things that they can do. So concept 17.6, comparing gene expression in prokaryotes and eukaryotes, reveals key differences. Prokaryotic cells lack a nuclear envelope, allowing the translation to begin while transcription is in progress. Here what we see is prokaryote DNA, and we see all along its length gene, this may be just one gene, being transcribed, the messenger RNA is being produced, and a bunch of ribosomes are already jumping all over the mRNA, and we see that in the color cartoon down here. So as soon as transcription is occurring, translation is occurring, and we see the growing polypeptides on the messenger RNA as it is being translated by the ribosomes. So the transcription and translation are coupled in the prokaryotes. In the eukaryotic cell, the nuclear envelope separates transcription from translation, so functional subcompartmentalization due to membrane-bound organelles. Extensive RNA processing occurs in the nucleus before the ribosomes can have a chance to interact with the mRNA. Concept 17.7, point mutations can affect protein structure and function. So there are mutations that can occur. They are changes in the genetic material of a cell, such as point mutations. They are changes in just one base pair of a gene. The change of a single nucleotide in the DNA's template strand leads to the production of an abnormal protein. So here we see wild-type hemoglobin DNA, CTT, and the messenger RNA, GAA, and the normal hemoglobin has a glutamine residue at this location along its length. In the mutant hemoglobin DNA, it is CAT, resulting in a message that has the codon GUA, and this signals for valine amino acid residue, where the glutamine, or glutamic acid, I'm forgetting my amino acid abbreviations. Glutamic acid is supposed to be. So a valine is substituted for a glutamic acid. So types of point mutations, they can occur within a gene and can be divided into two general categories: base pair substitutions and base pair insertions or deletions. So with substitutions, there's a base pair substitution, and it is the replacement of one nucleotide and its partner with another pair of nucleotides, and it causes what is called, or can cause what are called either missense or nonsense mutations. So here we see in this illustration the wild type mRNA with the protein met, lyse, methionine, lysine, phenylalanine, and glycine, and there's the stop codon, and the carboxy end is there. So with no base pair substitution, there's no effect on the amino acid, with a base pair substitution, in one case, there's no effect in the amino acid sequence. It just replacing the C with the U, but that still encodes for glycine. With the missense that we see in the middle here, there's an A instead of a G, and this does change the codon to code for serine rather than glycine. So there is an amino acid substitution as well in the missense. In the nonsense, what happens is you generate a stop codon that causes an early or premature stopping of translation, and you get a non-functional protein. So insertions and deletions are additions or losses of nucleotide pairs in a gene. So this can create problems because it's going to shift the triplet codons. Remember, it had to be 3 in a series. Well, now if you insert one in between 2 that are already there, you can see that you have a totally different code. So they may produce what are called frame shift mutations. So here we see the wild type messenger RNA and protein. Again, the methionine, lysine, phenylalanine, glycine at the carboxy end and the amino end to the left. So with a base pair insertion or deletion, you can get a frame shift causing an immediate nonsense. So here's a stop. A frame shift causing an extensive missense. So U is inserted here. So now rather than phenylalanine and glycine, getting leucine and alanine, and every amino acid, if this continued, just about every amino acid would be different in the protein. And then here, insertion or deletion of 3 nucleotides. There's no frame shift, but there's an extra or missing amino acid. Thus, we consider mutagens. Mutagens are of a variety. Here we consider spontaneous mutations, so they can occur during DNA replication, recombination, or repair. So in dividing cells, the DNA is susceptible to many environmental factors. So mutagens can act on the DNA during those times predominantly, but especially in the cell types that undergo mitosis and, of course, meiosis for gamete formation. So mutagens are physical or chemical agents that can cause mutations in the DNA. So what is a gene? Revisiting the question. A gene is a region of DNA whose final product is either a polypeptide or an RNA molecule. So we shift our way of thinking about a gene, one gene, one protein, to thinking of gene producing RNA, and RNA having a variety of functions, one of which is determining the amino acid sequences in a polypeptide. But still, we refer to a gene product as a protein. So the summary of transcription and translation in a eukaryotic cell. We can see the transcription. The RNA is transcribed inside the nucleus. Here's the RNA polymerase II. The RNA transcript, primary transcript, is produced. There's RNA processing. The RNA transcript, or the pre-RNA mRNA, is spliced and modified to produce the messenger RNA, which moves from the nucleus to the cytoplasm. The formation of the initiation complex will occur. You see that beginning to occur here. And then the amino acid activation is occurring with the amino acyl tRNA synthetase, and then translation occurs, initiation, elongation, and termination. So a succession of tRNAs add their amino acids to the polypeptide chain as the mRNA is moved through the ribosome. One codon at a time. When completed, the polypeptide is released from the ribosome. So this completes our discussion of